Good morning, dear students. Welcome back to the botany class. Today, in this session, we are continuing unit number 17, genetics, in which classical genetics, chapter 2. In the chapter classical genetics, in the previous videos, we come across Mendelism, who is called father of genetics. Why Mendel became successful in his hybridization experiment, why he chosen God and pea plant for his experiment, everything we discuss. Now we are continuing Mendel's experiments on pea plant. Mendel's theory of inheritance was known as particulate theory, which established the existence of minute particles or heredity units or factors. Nowadays it is called genes. Mendel performed artificial pollination, or also known as cross pollination experiments, with several true breeding lines of pea plants. What is meant by true breeding? A pure breeding or true breeding lines, it means that it undergone continuous self-pollination, having a stable trait or character inherited from parents to offsprings. A true breeding line, it means that it has undergone continuous self-pollination, having stable trait inherited from parents to offspring. Matings with pure breeding lines produce offsprings having specific parental traits that are constant in inheritance and expression of many generations. So what's meant by pure line breed? It refers to that homozygosity only, because pure breedings are always undergoes repeated self-pollination. We know what's meant by fertilization. The fusion of male and female gametes called fertilization, which produces by the same individual, that is pollen and egg, which are derived from the same plant, which is known as self-fertilization. Self-pollination, which takes place in Mendel peas. You know that Mendel conducted experiments on garden pea plant called Pisum sativum. During his experiment, Mendel removed the anthers. Removal of anthers from a flower, a bisexual flower, is called emasculation. Mendel removed anthers before fertilization starts and transfer pollen from another variety of pea to the stigma of flowers where the anthers are removed. I'm repeating. In bisexual flower in Pisum sativum, he removes anthers, then he transfers pollens which is collected from another variety of pea to the stigma of flowers where already anthers are removed. This results cross fertilization, or we called artificial pollination or cross pollination. This cross pollination, which leads to certain hybrid varieties with different traits. Hence, the Mendel's work on the study pattern of inheritance and principles or laws formulated which constitutes the Mendelian genetics. Mendelian genetics. Dear students, before the Mendelian inheritance, after Mendelian inheritance, if you observe, number of studies are made on hybridization in plants and animals. Mendel, which made foundation on it. The first model organism used in genetics is said to be that garden pea called Pisum sativum. In Pisum sativum, Mendel have been studied 7 characters of the Pisum sativum. What are the 7 characters? The 7 characters which he studied: first, stem length, in which 2 alternative forms are observed, 2 traits are observed which are contrast from one another, called tall and dwarf. The next, pod shape. Fruit of Pisum sativum is legume type or pod type. So the nature of the pod, inflated or constricted. The shape of the seed he selected for his observation was round and wrinkled. The seed color of the Pisum sativum was studied by him, yellow and green. Likewise, position of the flower, axil flower and the terminal flower, color of the flower purple and white, color of the pod green and yellow. All these cases, the 7 characters, 2 alternative forms are found. Mendel, who studied 7 contrasting characters in Pisum sativum, in which tabulated over here you can see the 7 characters studied by the Mendel, such as plant height, seed shape, cotyledon color, flower color, pod form, that is the nature of the pod or shape, flower position. Next, Mendel's 7 characters in the garden pea plant, which is shown in 7 chromosomes. You can see the 7 chromosomes, in which flower color and seed color is found in 1 chromosome, that is first. Third chromosome in which nothing, second chromosome in which nothing, fourth chromosome in which pod shape, plant height and flower position, gene for the flower position, plant height, pod shape is there. Fifth chromosome in which pod color, sixth chromosome in which nothing, and seventh chromosome in which seed shape you can see. So Mendel worked at the rules of inheritance and arrived at the correct mechanism before any knowledge of cellular mechanism. On or before Mendel's experiment, there is no knowledge about DNA, cellular mechanism of living organisms, genes, and chromosome became available. Only after Mendel, who is having insight due to his continuous observation, proper recording of the results during the hybridization experiments, mathematical calculations, meticulous work on the mechanism of inheritance, which play an important role, which lead to the development of improved crop varieties and a revolution in crop hybridization. Mendel died in the year 1884. After that, in 1900, the work of the Mendel's experiment was rediscovered by 3 biologists, Horal, Hugo de Vries of Holland, Karl Kahns of Germany, Erich von Schamack of Austria. They rediscovered the Mendel's findings and they found that Mendel's findings are correct. Dear students, when we study about the Mendelian history or experiments on the history, we can understand how an eminent work was conducted in those days without any modern scientific equipments were not available. Mendel's foresight thinking, meticulous work on the statistics which he prepared based on the mathematical calculations, observation of the results, very keen and proper recording of the results of each and every generation during his hybridization experiments, variation, differentiating the variations which is seen in the parents and offsprings are all brought a great success to the Mendel. Hence, he called as father of genetics.